Today we have a webinar, very informative kind of the webinar on the new genome technology, that is the CRISPR technology. And nowadays, this is one of the molecular technique by which we can edit the genome sequences, so by which we can, on the molecular level, can combat with the different kind of the diseases also. And in order to deliver this kind of the or to give information not on the theoretical kind of the level but also on the practical level, we have a eminent personality, we have a very a trained personality in this field. The person who is having the academic kind of the you can say that the experience also and the industrial kind of the experience also, Mrs. Sophie John, ma'am. And I will hello ma'am and welcome to PU family from the Department of Biochemistry and Genetics. I heartily welcome you. And as Shailaja ma'am is involved in some another meeting, so I have to take over this webinar. Okay, so I again welcome you in our Biochemistry and Genetics department. So firstly, I will briefly introduce Dr. Mrs. Sophie ma'am. She has completed her BSc in biochemistry and MSc in medical biochemistry. She has got gold medal in BSc biochemistry and she has completed her MSc from Sri Ramchandra Medical College, Chennai. She has work experience of teaching to the medical students of MBBS and BDS, and at present she is associated with the Tagore Medical College. She has industrial experience to work into the QA/QC department of a very popular industry, that is Biocon, which is based at Bangalore. Apart from that, she has experience of working in the laboratory associated with the clinical biochemistry part, working with these serum uric acid detection and its effect on the cardiovascular kind of the disease. She has the experience of medical content writing also, and with that she has trained so many kind of the people in quality auditing of the clinical laboratories associated with the handling of the material, the pathological kind of the material, and has worked with the industries also and has the academic kind of the experience also. So as a CRISPR technology, it is a very new kind of the technology. Initially, we are just involved in the molecular techniques as transformation, conjugation, and we perform it in the laboratory. But nowadays, CRISPR, like that of the whole genome sequencing, the CRISPR technology, it is very powerful. Though there are certain kind of the advantages and disadvantages are there, but still it is considered to be a very popular kind of the technique. So, ma'am has experience in working with the CRISPR technology, and not taking your more time in telling about CRISPR technology, I switch on to give the lecture talk. I hand over this talk to ma'am, Sophie John, ma'am. Okay. So over to you, ma'am.
Okay, thank you so much. And I am really happy and excited to teach this topic to the students and everyone over here. This is like, I like to even perform this for the non-medical science, or everyone can know about this, because this is related to the future disease things. So we have to be very precaution, and we have to like prevent the diseases for the future castings and all. So I really thank you for this opportunity to the dean or everyone for this PU family, and hope we can start the seminar. Okay, so hope the presentation is clear for you.
Yes, ma'am, it's clear.
Okay, fine. Just a minute. I have to make it in full size. Yeah. Okay, so CRISPR technology, the editing tool in the life science. So this CRISPR technology it is so powerful that you cannot discriminate or imagine how powerful it is. Can you ever imagine in your DNA you can cut, splice, and chop up the unwanted mutated thing and insert a new one. That is this process. How powerful is that, and that much excited it will be. So that the development of this CRISPR system is like invaded with an infected virus. It is taken up, and this is like mostly like with all the molecular techniques done with an E. coli. This technique is also done firstly in bacterial E. coli, and it is done by the two great scientists, the lady woman, the powerful Nobel Prizes of 2020 in the chemistry of Jennifer Lopez and Chapman. So these two people have got this technique via. I'm not saying they found it first of all. This technique is done in 1950s. This technique, the CRISPR formation, is like absorbed or like created from the 1950s. But this technique slowly, each wise, specifically proteins, DNA, everything slowly, they found to indicate each thing. And in this 20s only, this CRISPR is like widely or spreaded in the world. This name is so important. So first of all, CRISPR, though it is an editing tool in the life science, it is not only interested in the life science researchers, but also in the industrial biotechnology. Therapeutic drugs are needed, even for the agricultural use, and even for a common man if he is like infected with a viruses. Or another one detail I just want to indicate is that this February 8th, 2021, a patient of a sickle cell anemia patient named as Victoria in the US, she has recovered her full totally disease of a sickle cell anemia, which she had it in her lifetime process. She is like free with her sickle cell anemia disease and all. So can you just imagine how happy she will be? A gene is totally taken from her, and she is free, and she is living to live her furthermost life with her. So this CRISPR can make a new, even a new human can be created in that. Of course, we can fight the cancer, we can fight with viruses, even we can create a new humans, but that too with the ethical issue only. For right now, there's no issues and all. I mean, right now there is an issue. We don't have ethical process, but still in the future, this can really happen. This is a revolutionized medicine system. So CRISPR, this is a very mouthful, big name, right? So how can we like acronymically, we can break up is like the cluster of regulatory interspace short palindromic repeats. This is the CRISPR's elaborated name. This seems very big, but I can define you in the reverse way. Come, we'll go for the reverse thing. Repeats. First of all, there is something named as repeats. What does it mean? So here we have a picture of a prokaryotic cell, bacterial cell, and we have something like a bacteriophages or infected phage that's seen at the top of this bacterial cell wall, right? So in this bacterial cell wall, we have infected phage of bacteriophage that gives its DNA to the prokaryotic cell DNA, right? So just think, what is the DNA? Here you may think DNA, we can cut, edit, splice, do everything. So DNA is the very important thing. Never think so. DNA is just a target DNA we needed here. Because as I said, the infected phage which just inserts in our or in the prokaryotic cell, it gets injects its invaded genetic DNA inside this prokaryotic cell DNA. What happens is, just assume this is our DNA also, right? So what our DNA says, we have an adapting immune system, right? We have, what happens if you get a fever, cold, or flu, or anything? We invade that. We take up the viruses. How? Because the DNA thinks that is our particle only, and it takes up. And slowly, this DNA sequence becomes sick, and we get like infected. Right? The same thing. But when we get infected also, we have some antibodies in our body to defect the foreign particles, and we have white blood cells to invade it out or just flush it out, go out from here. And we have our immune system very like precautionally waiting to start or fight with them. Same thing here, the bacteria is also having its adaptive immune system. You won't think it has its own adaptive immune system. So what it does it also takes up, but in a process, what happens? This foreign particle keeps on coming, and our immune system or our weaker. At that point, DNA accepts all the genetic people of the invaded or like infected phages and all. Then DNA takes up, and it gets infected, and we get lots of mutated formation. We have a proofreading of different variation. We fall into a different frame shifting mutations and all. We cannot chop, and we get a disease formation. But this CRISPR is so important and powerful is that it already has a CRISPR array over here. So I just want to just indicate you, it already has a CRISPR over in this area, right? So it has a CRISPR array. I'll say you what it is. And we have something named as Cas proteins. This is the Cas proteins, cascades, and we have the Pam sequence, and we have the foreign DNA, which I told it is given from this bacteriophages, and we have something like spacer, right? So these all are the very important thing that you should know for this CRISPR formation, right? So what else is there? So we'll come for the topic or the abbreviation of the cluster of regulatory interspace short paramedronics repeats. As I said, it is having something repeats. What is that repeats? Nothing but our DNA. DNA totally is a long stretch of DNA. You cannot like imagine how big it is. It is a millions and trillions of, that is present as a thin filament in our body. Just skip to this. We'll go for the introduction first, right? Okay. So genetic material. What are the genetic material which we have in our body? Of course, our DNA and RNA. So genetic material is nothing but the substances that carries the information determining the properties of that organism. So here the DNA is a genetic material for almost all the organism. In this, we have to particularly know about the directions of DNA and RNA, how it is going out, right? As I said, DNA is not only important over here, the RNA plays a major role. CRISPR is nothing but the handful of RNAs, right? So we have the direction of DNA from 5 prime to 3 prime direction. That is the template strand where we really need it, right? And next, the RNA. It is a genetic material for bacteriophages, viruses, and some plants even. Of course, we have it in our human species also. But this direction is from 5 prime to 3 prime in this direction, right? We have the arrows indicating from 5 prime to 3 prime direction of DNA, and again the opposite from left to right, that is from 5 prime to 3 prime in mRNA. And other strand of DNA, as DNA is a double helix, we have the other strand that is also sensing from left to right only, 5 prime to 3 prime as a direction of RNA. We'll go for the next thing. What is a gene? Right? These are basic concepts we know. It is very easy to skip up through the other slides and know about the technique very easily. So gene is a functional unit of a hereditary which carries information from one generation to other generation. So we get the elaboration or like someone saying, oh, he's having diabetes, of course he'll get it for the next generation for his son or like that. So this is one sort of thing to explain what is a gene. So gene carries information from one generation to next generation. Of course, there is many new futuristic therapies to cleave even the diabetic for the next generation also. Through this CRISPR and all, we can do it. And next, chromosomes. What is a chromosomes? It is a linear end-to-end arrangements of genes associated with proteins. So there is a linear end-to-end arrangement. And end up with the proteins. There is nothing but our transcription to translation. This chromosome is going to help us to give up the end association of a proteins. And we have a genome. It's a sum of all the genes of all different chromosomes of the cell. Now we'll go for this picture. We have a chromosome, we have a chromatin, and just think how two telomeres or a centromere and all of a chromatin of this linear gene from end to end point gives us a thread filament and ends up in a base pairs of DNA. Can you ever think chromosome is so immersible, beautiful thing that it can give us a DNA to the end. So just it has a two chromatids and in one coil can give you the loop of nuclear scaffold things, and this scaffold can give you a twisty form of one loop, and it is a tight fiber thing, it contains of packages of histone proteins. So In this, it is very elaborated to know how our DNA is seen. So the chromosome is the major thing in our cell that is like packed with a or made up with a chromatin layer, and this is a condensed fiber of a six patterns, and it's condensed with nothing but a very tightly packed histone. Proteins are over here tightly bounded and twisty, and these histones are said to be a totally in a bunch of nucleosomes, and this gives a DNA in a very thin layer in the end when we like separate each layer. So DNA is the end product, right? So as I said, where do you see this mRNA and all, or RNAs totally? So in this, we have first thing we transcribe, right? We are transcribed with mRNA from the DNA. So DNA is being transcribed to mRNA to give us a protein, right? So this is our normal process in our cell to be replicated by a DNA, and transcription happens and translation happens, and we get a protein where it is needed for our survival. And wherever the proteins are needed, our liver gives up and we are like taken up in it and we survive with that, right? So DNA plays a major role. But what is the role of this mRNA, particularly mRNA? mRNA takes the messenger of the DNA totally and gives it directly the information of this RNA, and it gives the amino acid as an end product, right? Amino acid as an end product where we need it for our survivals and all. So why we need the amino acids and all? We need for our translation process and for our nucleotides. We need it because without this nucleotides and all, we cannot survive, right? So next, now this is the direction which I want you to really notify. So this is our coding strand. This sense strand of the DNA. This is from the direction of phi, or from I can say from left to right, that is phi dash to the 3 dash, right? And we have the other strand, which is template strand of the DNA. It is having this strand of 3 dash to 5 dash. And we have a complementary strand or an mRNA from 5 dash to 3 dash. Hope this is clear for you. Fine. So, as I said, mRNA are nothing. It is the DNA to transcribe into a RNA molecule, and that process had to be transcription. And our mRNA is a template to serve for the protein synthesis. Further protein synthesis should happen with the help of this mRNA through the process translation. And our mRNA transcript, that is a single strand RNA that encapsulates the information containing in the gene. And RNA, it might be smaller and it is very mobile. It keeps on moving here and there. This mobile RNA is nothing but it is having the same sequence of the DNA also. It is built in a form that contains the same information of a DNA. Okay, so before going on to this, I just want you to show you or tell you first of all, what is a gene knocking out? I hope you have listened this name, gene knocking out, right? It is nothing but a repressing the gene to make the gene stop. No, you cannot function over here afterwards. That is the only thing we can say it in a similar words to have a great elaboration of this thing. Gene knocking out. This is really very important in the CRISPR. So this is done due to like when our double-stranded DNA breaks up, right? How it breaks, I'll show you that. When our double-stranded DNA breaks up, right? It breaks up into two formation. And then we have something like a ligase, our enzymes that goes and breaks up, and the mutated is taken out, and the other two ends is going to join. So hope you think the mutated part is taken up, so the frame or the reading everything goes correctly? No. So there'll be something like in a non-functional protein is going to happen over here. So when you insert something also, we need a proofreading in a correct thing. So anything that is mutated and taken out and joined the DNA sequences togetherly with an inappropriate sequence of reading or any addition of insertion or deletion of proofreading mistakes or blood transfusion can give up a gene knocking out. Blood transfusion, that means wrong transfusions, right? So that can give up a gene knocking out. So anything that should be like correctly done is like when a mutated part is taken also, we have to insert the correct one. Then only the frame will be like correct, or the reading of the process will be correct. So we should really know about this gene knocking process also. So this is the one thing which I really want to explain you. Cluster. of regulatory interface, short palindromic repeats, right? So till now the basics is done. So here I just want you to say something is said to be repeats. What is this repeats? We have the black things over here. This repeats are nothing but our DNA sequence of 20 paired amino acids, okay? Or base pairs. Now, we have an infected macrophages or something, okay, of our virus that comes inside, right? It comes inside, it sees what it does in the first image, it incorporates inside our DNA, right? It comes and injects inside our DNA and DNA accepts it, right? Before that, I just want to explain this. Something repeats is there. I told you the black blocks are the repeats, nothing but a 20 base pairs of our DNA. Short, they're very short. We cannot take 100 base pairs and all and then cut it and read it and mutate it. We take very short pairs of DNA and then we recorrect it, right? So this is a short and it is like repeats. And what is palindromics? Palindromes are nothing but you have to just remember that whatever the length of this DNA is there within the 20 base pair or within the 40 base pair, not more than that. So within that base pair, the reading of 80 GC, our genes or genetic codes, that is going to like similarly seen identical, and well, in which direction when you read it, like from left to right or right to left, the sequence is totally clear. There will be no odds or no evens. So palindromics nothing but the sequence is very identical, and when you read from right to left or left to right, there will be no corrections or like no mistakes. It's all clear. So it is that sequence is like palindromic. So till now we learnt about what are the repeats we saw over here, the DNA, and the palindromic. In which sequence we read, it is like correct. The manner is like same, and it is short, right? And next is it is interspaced. What is this interspaced? Yes. So I told you before, we have some Cas enzymes. This is a protein or enzymatic protein which is really neat for this CRISPR technology. And the space, these all black blocks are the DNA. And what is this colored one? Blue, yellow, blue, green, something. These are the spacer DNA, right? What is the spacer DNA? Yes, I'll tell you now. So total this black blocks, blue blocks, everything, this formation is said to be a CRISPR locus, right? This region is said to be CRISPR locus. Now, what is in spacer DNA? Spacer DNA is nothing but in this CRISPR, particularly in this CRISPR arrays, this is CRISPR array, right? This CRISPR array, spacer DNA is nothing but whatever the infected virus that is like invaded inside our DNA, this spacer DNA is going to take up this virus. Why should it take up? This CRISPR spacer DNA is nothing but it is a memory process. It is having such a memory that whatever the infected virus or foreign bodies comes, it is already having a duplicate copy of it. So this spacer DNA can recognize in this CRISPR locus area, this is a CRISPR array and this is a CRISPR spacer DNA, right? This blue colors is a CRISPR spacer DNA. So this CRISPR spacer DNA already knows this virus is going to attack us and this infection is going to come for us, and this flu or this bacteria or any disease is going to come inside our DNA. This CRISPR can really know it, that particular new spacer DNA can know it. So it is already having a duplex copy of it. So this infected virus, it takes up this spacer or the new space DNA takes up. Why? Because it's already having a copy of it, and it will take up, it recognizes it very well, right? It takes up and at the end it chops in the CRISPR array. So this is the very, very interesting thing, that our CRISPR takes up the virus or the foreign particles that comes inside our cell, and this new spacer DNA takes up because it's already having a copy of it. It knows this flu is going to come with this virus, so this virus, I'm already having it. You come here, I will attack you and I will not leave you away from this CRISPR locus area or CRISPR arrays. And we cannot, and this, apart from this CRISPR, this virus cannot go to the next DNA and have a sequence running out, right? So this, in this CRISPR locus, within the CRISPR array, our virus, whatever it is coming, or the foreign particles, this CRISPR takes up the virus into the new spacer DNA and chops up into that with the help of some enzymes and all. I'll let you know. So we need the particular enzyme here is a Cas enzymes. Okay. So hope till now this description is very clear. We have a repeated sequence of DNA as a black blocks, and we have a new spacer DNA also. That is nothing but it is a duplex copy of the foreign particles that it takes up and chops up within those sequences, and it will not leave it to the other sequence, right? So the CRISPR-Cas9, yeah, we have a Cas gene over here. We have 9, we have 13, we have 12, we have many numbers of genes over here. I mean, the Cas protein genes. So particularly our scientists of 2020 Nobel Prizes, two scientists, they have choose the CRISPR-Cas9 process. So Cas9 is such an important in our 2020 thing. So CRISPR-Cas9 based genome engineering technology will increase our understanding to disease processes and their treatment in the next future thing. So adapted from the bacterial adaptive immune system. This is totally adapted from the bacterial thing that we are going to work in our eukaryotic process. So the top world's development of the gold standard genome editing technology is the CRISPR-Cas9. The gold standard technology is CRISPR-Cas9. So this is the history, as I told, not only in 2000s, this has been identified, this is done not even in 1987, done in 1950s. But in 1987, they reported first as a CRISPR repeats and all, and they identified the Cas genes, then the proposal of the defense mechanism system, and around what is a Pam sequence, everything will be like coming in the next upcoming slides. So this is the history. Totally in 2000, like 11 Cas cleaves the DNA in bacteria is being like produced or approved. Cas9 cleaves the bacteria. How it cleaves, we'll come and see it. And so this is totally the history. So from 1987 to 2005, we get the specific RNA processing is there for the CRISPR. And then in our 2010, we have the type 2 CRISPR, Cas cut the DNA. We have types, many types are there. So totally we'll come to the end that till how where we found this in 2018, the first human clinical trials is like taking over for the beta thalassemia patients. That is not the in vivo, that is an ex vivo. So till now, none of the patients are directly like given up inside, but the process is happening by taking up our serum outside or the blood outside, and the process done in the laboratory. But this February 8th for 2021, a Victoria patient of sickle cell anemia, she has given up her DNA, and it's like if she has injected it inside the CRISPR system, it is totally cleared the sickle cell anemia. Fine. So this is a journey or the use of the CRISPR from the DNA. So where and all the CRISPRs are used. This development of CRISPR system, it is so therapeutic, so revolutionary medicine that it's been done in animal models and then genetically variations and materials like crops, food products, drugs and diagnostic systems, gene surgery, therapy, and drug developments. Everywhere this CRISPR is like utilized and done and up to come. You won't believe per day or per week you could get like 100 of journals of the CRISPR that's updating new things are keeping on coming. It is like enormous thing. You cannot just imagine. This CRISPR plays such a big role because it is the only thing for the CRISPR is to it targets the spacer DNA or the targeted DNA with the infected virus or bacteria, anything is there. It goes and cleaves up and we are free from the disease. And in future, of course, we can really create new things, new human. And I don't think whether you have got this news and all. In 2016 or 17, a person, not a person, I can say a person, he is a PhD of biochemistry. He itself did a CRISPR technology. He injected in his body and done it in his home. But this is ethically approved and all. He's like jailed also for 2-3 weeks and all because this is like not beyond done with the ethical procedure. He's done it by itself, but he's like he's living. Please don't try this. This is an information. And the CRISPR types. Now we'll go for the basic thing. CRISPR types having a key effector proteins and 6 types of CRISPR-Cas. As I you show you, as you have seen in the classification types of type 2, type 3, type 4, so those types will be seen here. So this is the effector protein that really needs for this CRISPR-Cas9. Nothing but our Cas9 and our guide RNA. Okay, so Cas9, what is Cas9 protein? This protein is nothing but it is having a domain of 4 to 6 types, right? This Cas9 can bind our target DNA where it is like mutated and all. It targets is nothing but the components of two. One is the crisp RNA, that the CRISPR of our RNA, and a trans-associated CRISPR RNA. These two complex form together to give us a guide RNA. What are those names? The name seems so different, right? And very anomalous and differently. So we'll see what it is. So the Cas9, this is a process totally. Just check out the process, how the CRISPR is done. So Cas9 and our guide RNA, it incorporates together and it enters into the targeted DNA where it needs. And this hairpin loop like guide RNA, it likes complement to the target DNA, complements. And if it finds any mutated, if no, it is finding any mutated thing in the complementary or the target DNA, what happens is it chops or incorrect the gene by cutting it into 2 slices and leaves it apart. And the mutated one is like chopped and taken up, and we can insert something inside also within and close it up, right? So there is an here when it chops itself, that minute itself, the trigger of an innate cellular repair is like going to peep up and say, you have chopped up, I have to insert and delete, and here we can get the gene knockout systems, right? So this thing totally in the red color and yellow color is totally incorporated to give us a Cas9 protein. Now, how the researchers are in the normal laboratory for our bacteria, how we like grow up and say there is a gene knockout system happening. So with the plasmid nothing but we have a Cas9 that is with the help of a guide RNA. These two incorporates with the help of a guide RNA. We take the bacteria of a LAC-Z. Okay, this LAC-Z bacteria is from the streptococci. We take it, the LAC-Z that produces the galactose or lactose, and this gives us, we form colonies with the help of our restricting enzyme antibiotic resistant gene. This all together plasmid, we plate it up in bacterial growth media. With the help of the growth media, we apply our plasmids in the tube and then plate it in the petri dish and keep it for the incubator. And before that, we have to spread it very well. This is a normal process which we do it in our lab thing for school lab or practical labs or college labs. So they spread it all over the area of the plates and keep it overnight in an incubator of 47 or 37 degrees Celsius in the room temperature. And then next day we have to check the results. So what will be the result? Of course, when the bacteria and lactazine enzymes are present, we have a lactazine enzyme of the bacterial growth, and also we can see the blue growth thing or a blue bacterial colonies. So this is a question for you. As I told, that there is no null, but these all process can be happen. And in the bacteria, this is the process where I said to say the bacteria that gives a LAC-Z enzyme gives up the production of beta galactose, and this beta galactose releases the X-gals, that is a white crystal that should be really incorporated out in the formation of the colonies to give out the pigment of blue color, right? So this is the process we found it in the colonies of our petri dish, that beta galactose that forms should really give out the X galactose out. Otherwise, this process is like going to nil up, or like we have to give a gene formation, or we have to like insert some gene therapeutics to give out the X gal outside of the for the blue pigment to be arising. That we are going to see up over here. So in the bacteria which does not have the presence of LAG-C enzyme or gene for it, so what happens? It will not release its beta galactose enzyme, and the X gals cannot like come outside to give a pigment of blue pigment. So this is a white colony. So this tells us that the bacteria with the function of LAC enzyme gives us a blue colony or a blue bacterial colony, and the disable of LAC genes. So this gene is like mutated one. Our Cas and our guide RNA is going to like incorporate it and chopped it out. So what happens is we won't get the colonies out. So this is a mutated DNA that can give us a white colony. So in this, we can say the presence that this mutation is going to happen in this in this bacteria, and we keep it away apart, and we will cut that sequence, and the next other sequence will have a correct manner of our basis. So this is the thing. The blue color colonies is said to be no knockout. That's a perfect one. And in this, we have already knocked out the mutation of the LAC. So hereby, today we discuss about the some of the Cas proteins we saw. So furthermore, what are the Cas proteins and the function, and what is a Crispr RNA and trans RNA, and what is a PAM, and how we visualize our Crispr RNAs in the laboratory. We'll be seeing it. So I just tell up what is the main thing for the Crispr is that our signature protein is the Cas9, right? And we need a guide RNA, as I told it already. And next thing, for our CRISPR types or the CRISPR-Cas system for genetic engineering, for a proper identification of, we need a 3 accessory components. One is a leader sequence and precursor CRISPR RNA sequence and PAM sequence. This is totally this much only. CRISPR just needs 1, 2, 3, 4, 5. 5 things, that's it. And with the help of our DNA, right? So everything when inject together inside a DNA with the correct process and steps, we can cleave our mutated and we will get the perfect DNA with no disease, right? So Cas9, as I told, Cas9 is not just a Cas9, it is having 6 domains into it. Each domain incorporates and does its function. So as I told, Cas9 binds the RNA and targets the DNA. So who binds the RNA is that our REC1 domain of a Cas9. It binds the guide RNA. And next we have the arginine-rich bridge helix or a bridge helix. We have REC1, REC2, bridge helix, PAM interaction, H&M, and the Rubic domain. So in this, REC1 is giving the full process to bind the RNA, because the first step is Cas9 and RNA, guide RNA should like bind it. Next, this 2 thing is going to get and cleave or like target our DNA. So who's going to target it? Who's responsible to like help out in the Cas9 is the bridge helix. That is our arginine rich. So it is having a rich of arginine of bridge helix that is initiate to cleave upon the binding RNA. And next we have the 2 H and Rubic C domain, our nucleus domains to cut the single strand DNA. So who's going to cleave in both the sides? Our HNH and the Rubic domains of a Cas9. So this is the process or elaboration what is a Cas9. So Cas9 totally is having a 6 domains. That is only slowly each and one is going to incorporate and does it function. And next one is a guide RNA. As I said, guide RNA is nothing but it is an RNA. It helps to target our DNA and give up the complementary sequence together, and it reads up the thing in the sequence and it identifies this is mutated and it cleaves it with the help of a Cas9. It cleaves up. So this guide RNA is a single stranded RNA and it is having 20 base pair longs. It is a T-shaped, right? It is a hairpin loop T-shaped one. And the guide RNA engineering is starting from the 5'N to 3'N to complementary to target the DNA sequence, right? So what is the leader sequence? Here I told you, right? Cas9, guide RNA, and leader sequence, and precursor or CRISPR RNA, and something we have like trans associated CRISPR RNA, and our PAM. These all the five things are really needed for this process. For a workshop also, we really need this. So precursor CRISPR RNA is nothing but a repeats of the CRISPR array. This is like as we have a promoter at the first side, and then we have our genome systems and our proteins. There we can see our leader sequence and CRISPR precursor RNA. So this is the one thing. How the process is that I told you already. The infected viral comes inside as a foreign particle in our DNA. So our DNA that is having a new spacer arrays, that is nothing but the CRISPR arrays, that adapts the foreign particle of DNA virus. It takes up, it adapts, and it holds up. And next, what is the expression going to happen? Our CRISP RNA or our CRISP cRNA, that crisp RNA is going to express on that new spacer RNA, and since you are my founder, come, the virus, come, you come, I will see how I can give. It expresses by giving out the knockout, gene knockout. And next is our third step. What happens is that CRISPR interferences. What is an RNA interference? The same process is only going to happen in CRISPR. Interference is nothing but it gives a non-functional protein. It eliminates or cut the two double-strand DNA breaks up, right? When the mutation is found, our guided RNA and our Cas gene 9 cuts the DNA, and mutated part is taken up, and some two ends are going to join together, right? The mutate is taken out, we have a space over some two, that two ends are joined together. But that protein is not a correct protein. The sequence will not be correct. If we insert or delete also, something is going to be enormously different in that DNA sequence. So what we do is we cleave that totally, that 20 base pair sequence, we cleave it, or we tell it like be silent, or that is silent is never knocked down, or they will say you just don't function at all. That is our knockout, right? So non-functional protein is found in that sequence, and that is done by the RNA interferences step. So these three important steps are the CRISPR-Cas9 system. It adapts the new spaces DNA and insert into the CRISPR array, and it expresses by the CRISP RNA to go and cleave it, and then in the CRISPR interferences, that DNA sequence is going to chop and say you are incomplete. Please don't work for the next process, and you are non-functional protein. So this is the classification of CRISPR-Cas, not Cas9. It is CRISPR-Cas. There are many types: type 1, type 2, type 3, type 5, and type 6. So in the type 1, first signature protein is Cas3, right? And next in the type 2, there are two again. And just I found to left to say this. In type 1, we have a subdivision of A, type 1A, B, C, D, E, F, and new upcoming thing is U is also there. I have not given here. In the new update, you can see it. And the first signature protein is Cas3. And in type 2, we have type 2A and type 2B. And this Cas proteins, which is all the signature proteins, are Cas1, Cas2, and Cas9. Our scientists have taken the Cas9 of the type 2 CRISP-Cas system, right? And in type 3, we have a type 3A and type 3B. And here the signature protein is Cas10 signature protein, right? And in type 5, we have Cas12. And in type 6, we have Cas13. These 13 and 12 Cas proteins are newly updated, taken up from the study of 2019. And the development started in 2018. So this is the process. This is the type 1, and we have the first signature protein, Cas3. Here we have the Pam sequence. Now I have to really say what is a Pam. Pam is the very important thing. It is the cleaving process where it triggers or gives a signal that I am mutated, right? So this Pam is nothing but it gives a sequence of that DNA region and tell or prick, give a signal, I am mutated. It even changes its colors. So in our machine of our CRISPR, it when we read it in our digital formation, in the sequence goes on DNA, ATGC5, and everything runs out. The PAM sequence in the machine, it really indicates and give us a sequence of colored or a signal one to tell us we are PAMmed, and it ejects and takes out, right? We can directly go and prick and cleave it. So we have our CRISPR RNA, or we say it as a cRNA. This is that loop over here, and our signature protein Cas3. This is the oldest model. They do it, the first signature model. Then the present model is that here we have our DNA sequence 5' to 3' and 3' to 5', and we have our CRISPR RNA, that is cRNA. cRNA is over here, and we have a Pam sequence that tells us I am over here, cleave me, right? And this in type 3 we have Cas10. Here we don't need this CRISPR RNA or trans associated RNA. We have the complex of Cas and we have the mRNA, and PAM is over there. It tells cleave me, and CRISPR RNA will be like associated in next step, not in this step, right? So in type 5 Cas12 protein, what happens is we have only the CRISPR RNA, and we don't have a trans RNA over here, and the PAM is detected over here. And Cas13, it is totally different. Cas13 is a protein that cleaves the mRNA also. This is very interesting, Cas13. I'll tell you the next slides. So in a very brief description, type 1, as I said, Cas3 is a signature protein, and you have the types over here. Till type 1, use the famous one, other than the new one. And type 2, we have Cas9 as a signature protein, and type 10 and type 3 Cas10. So this is the thing which I explained here. So how we see it in our projected one, Cas9. We said Cas. It's just a block like a ribose comes and attaches and does a translation process. No, it is crystallidally locus process of like this. It shows like this in a 3D formation, and it needs a Cas9. And this Cas9, our scientist has taken from the or the bacteria is that from Streptococcus pyogenesis, right? Here the mechanism, as I showed, we have a PAN and a CRISPR RNA and trans RNA, the hairpin loop, everything. It triggers the PAM with the help of the PAM triggers over here, and we cleave the process. And we have such other studies also over here. The new study, that is a Cas12 one and Cas13 one. Okay, so these study slowly in each mechanism. You can see that we have our Cas12, we have our Cas12, we have our Cas12, and slowly you can see that we trans RNA is there, CRISPR RNA is there. Slowly in a Cas3, we don't have this CRISPR RNA. This trans RNA, we don't have it over here. In the Cas13, we don't have it over here. But we have it in the rest Cas12 and all. Fine. The mechanism, again, I'm going to recollect it for you. A phage that inserts in our DNA. Slowly, the new spacers will eject or take up the viruses inside through the new spacer. We have our Cas genes over here. We have Cases, many Cases over here. Which Cas helps out? It survives or inserts it over there. And we have a trans RNA also along with the Cas proteins. And this new spacer takes up the thing, and next it what happens is it will cleave over here itself. It chops up, and see with the PAM sequence, it chops up, and the repeated other sequence go directly in a correct formation. Fine. This system demonstrates the incredible diversity by the presence of unique Cas genes and the mechanism of CRISPR RNA biogenesis, and it targets the mechanism and cleaves it. So this technique can be easily implemented in any lab or any space. We get these all equipments or like parameters, the bacteria, the Cas and the trans, and our newspaper, our new spacer, CRISPR, everything we get it in an agent company, and we get this parameters along with it, and we can proceed it in our PCR and fluoro machine and gel electrophoresis, and we can do it in our paper strip model also. These all are updated new technique. So hope this is very clear for you. This is a CRISPR formation, how it does. Adaptation module, where it adapts the foreign DNA, takes up its viruses and goes to the CRISPR array and cleaves over here. And then we have the pre-CRISP RNA. What happens is that this is the CRISP RNA. It is having also the spacer RNA, that is, this is like in zoom in, we can see this, right? The transcription happens over here. Normally, our mRNA takes up the messenger with the spacer, and this is slowly going to cleave off again. We don't want this spacer, right? Because it is infected one, virus one. It cuts over here and it gets interfaced and says, you just stop your work, and it cleaves off. The foreign DNA is degraded, right? So this is the one final thing I want to just show you. We will be in this cut process, what helps is our Cas9 and our guide RNA both incorporates, and the target DNA comes over here. And then we have a cut site with the help of our Cas9 domains. It shows where is the cut site, and our Pam is also going to help out over here, right? Our Pam says, cut over here, I am here, just cleave me. And then the cut process starts up. So our DNA is totally like double-stranded bricks. Just assume this is done in prokaryotics, but we the eukaryotics process also going to happen the same. And next, this is the repair machinery. What is that? Our when this gets break out, our innate cellular response or the repair is going to trigger out and say, just join me, and triggers out. So who is going to help out a repair machinery? The homologous direct pair, or this is like the, so these are the 4 things that is really needed for the gene editing. Homologous repair pair is also there, and we have our non-joining thing. Repair is also over there. So the blunt and a sticky end process is really needed over here to join our sequence, but it gives a non-functional protein only. Whatever the repair machinery joins or inserts or deletes or totally say give up a frame, give up our DNA sequence also, this DNA is going to, if it reads well, it is a correct DNA sequence, or else it is a non-functional protein sequence. So first, the same thing, CRISPR-Cas locus. Locus is nothing but it is having an adaptive immune system, the repeat sequences, and it is having the spacer DNA, and it is having a memory, right? What is that memory? Viruses come inside, I will capture you and I will trigger you. That is the process where the CRISPR locus is going to do directly. So this is a CRISPR locus. Same thing. This is a CRISPR array, the leader sequence, and you have a Cas genes over here. Cas genes, leader sequence, and the repeat sequences of our DNA. And 1, 2, 3 colored particles is our spacer DNA. And this is the totally this CRISPR array over here. And totally we say this is as a CRISPR locus. So now we'll go for the technology. What is the technology? We have to compound system to cleave the technology. With the DNA, RNA, we pair and determine to edit also, and it is a multiplexed complex, this Cas9. So we already saw how the CRISPR or a CRISP RNA is going to separate itself. We get the cleave of this separation of colored one is the infected virus of DNA, and this is our loop of our RNA. So this is going to like chop up and give it in this formation. So this is just to elaborate what is trans RNA. This is the bulge of our mRNA, and this is the lower stream of our mRNA. This is the spacer DNA, right? And next, this is also the same thing. This is the array where we see it in our machine process. So protospaces we will see here, or a new spacer. The PAM indicates over here in the red color. Wherever the mutated is there, nearly we found a PAM, and this adapts over the system over here. And in the expression, it cleaves it, and in interferences it gives an addition or deletion. A mutated one is over, like it adds up, but this protein is going to be non-functional. So here, this is our human gene thing where they have done it in a laboratory with the help of our Streptococcus pyogenesis Cas9 and Streptococcus RNase and trans RNase done. So this is the thing where we can see our human genome is correctly done. Here the proofreading of our gel electrophoresis, but you can see two other prints over here. That is nothing but the mutated one. We have to edit or delete it. Okay. So there's a Pam sequence. It chops up, right? So how we do it with the two things. The blunt end is going to help out. That is the homologous directed repair is going to help us over here to give up a new editing process when it is chopped up, or else a sticky end, the non-homologous end joining, right? This is going to step up to give us an indel. This is said to be a non-homologous end joining. So this is like knockout process over here, and this is a knock-in process, right? So now we came to a very special thing, the laboratory techniques. There are many tools. We have a detector, finder, Sherlock, chips. Now we have a small chip. Just take your DNA sample, put a drop as the HIV code is there or our pregnancy test tip is there. Like that, we have a small chip erased. This test is done, but it is not ethically like come up to the outward procedures. So we are waiting for it, and Sherlock is done, Crispr detector and finder is done, and Crispr recognition tool is there. So many things we can do it in this laboratory technique. First, what happens is this is really easy one, time reliable, fast results, efficient and cost efficient, and this is easy to program or reprogram, and you can create lots of libraries. That is our DNA libraries or cloning or cell line, right? So this is very important, and you can have a tracker and reporter also. What are these trackers and reporters? We'll see it later. So this is the knockout experiment, the first experiment. Of course, in our mice, we do it for an embryonic stem cell. In those days, like it takes around 1 to 2 years to knock out the embryonic diffused region where to cleave up the DNA or the mutated DNA. It takes like 1 to 2 to 3 years. But this, whereas this Cas mediated, when it comes up, we can get it within a month, the result. Right? Might be not within a month, might be within 3 months, not beyond that. We get the results. So that is far like efficient, and that can be done very good for this knockout procedure. So CRISPR technology is intended a variety of additional organisms genetically trackable and inducing parasites. Right? And I really forgot to say this. In China, a scientist have like done it for embryonic twins. They have done this CRISPR technology where those two embryonic babies or fetus is like affected with HIV. That scientist has like done it in an embryonic stem cell itself. They did that CRISPR procedure, and they have cleaved the HIV viruses from that embryonic fetus. It's so lovable and excited to know it. So they have cleaved this process also. So HIVs also can be cleaved. So what are the other things can be done to cancer, wound healing, synaptic transmission, and T cell differentiation? Everything can be like recovered with the CRISPR and eliminate our disease. So the successful edit human embryos in the laboratory is going to happen, and this can be this is like totally happen. You can say it happened, but not in directly in vivo. It is done it in ex vivo only, right? So, as I said, cancer, wound healing, synaptic transmission, and circadian rhythms, everything can be knocked out with this CRISPR-Cas9 system. And with the model of a pig model, that is a hunting disease, in that what happens is we can like transform or like we can change the organ itself through this the CRISPR technology. So that can also be done with this pig model. They have done it. So in future upstanding, we can see it in our humans also. So CRISPR-based screening has applied in a large number of biological questions. You can just ask why we get a mitochondrial disease, why we get a DNA repair, why we have a cell-fate specific apoptosis. These all things can be like cured, but everything is going on slowly. The research is really going, as I said, per week you get hundreds of updates of our cancer or upcoming diseases. This CRISPR helps in everything. So this is a revolutionary medicine area with the therapeutic drugs and all. We are really going to finish off our upcoming or new upcoming diseases, and we have a new human growth also with a clear, clean, and crisp. So, crisp mediated gene lockdown has been applied in many things. You won't, this is just, we talk about the prokaryote and eukaryote. Have you ever imagined agricultural crops like rice, barley, soya, maize, wheat, tomato, potatoes, lettuce? These all things are also being incorporated with the CRISPR technology. What is that? They like grow very healthily, and they should be not drought resistance. There should be increased yield, and the time also should be saved. So these things have been done in the biotechnological life of agricultural crops, and they have really approved it. And this is the one important thing which I if that is this is a new case like done recently only in this 2020 or 2019 only. They have found this cas 12 and 13. They have implicated in the process of an agricultural process also, and also in the human process also. So Cas12 has approved a base editing approaches that applied in a number of plants, maize, tomatoes, potatoes. And now, what is the Cas13? Cas13 is really very important to know how our RNA interfaces is happening. Same only in our Cas13 is also happening. What happens is our Cas13 cleaves the mRNA also. How come it cleaves the mRNA? Is that mRNA also keeps on going, right? It should also have a stop. So there will have an interferences of mRNA. So this Cas13 is going to cleave it or stop it and can do it. So the advancing human health over here, as I said, the transplantation into human patients and Zika virus, Ebola virus, this Cas13 is really giving a good thing. So we have some Sherlock platform. What is it? Using a gel electrophoresis and using a fluorometry and using a strips, we can identify or stop our RNA targeting reporter. So we have an isolated DNA, we take it from our blood, urine, and saliva. We double strand the DNA break and we take up the RNA. Also, we stop that or we amplify it and take the target sequence and we incubate it in our thing, and we lock it. We have to lock the reporter. Who is our reporter? mRNA. We have to stop it. It cannot just go on and do its work. There will be silent promoters also to cleave the mRNA. So the bunch or the cluster of mRNA can cause also a disease. So we have to lock our RNA also. So at that period, we need to use this system known as Sherlock platform. That's nothing but specific high sensitivity enzymatic reporter unlock. So this Cas13 triggers our mRNA and we report it to an unlock system. Through the gel electrophoresis, we can plate it and see our logged or the cleaved one and uncleaved one, we can report it. And through the fluorometry sense, we can see the fluoropore that is like added inside it and cleave it. And to the strip paper, we can see that we can cleave our mRNA also, see the rating, whether it is in a negative or whether it is in a positive strip. So if it is a negative one, so we can say that our mRNA is good. And when it is like in a positive one, we get another one band that is collected directly. All the mRNA is collected and give the uh, strip, and the second layer line is also seen. So recent updates, as I said, the Cas13 based diagnostic results is also being lipolysed and easily reconstituted to the addition of water. So with the help of water and our fluorometry and our gel electrophoresis, we can do it. And we don't need a requirement of a refrigerator is also something some scientists have discovered in 2017. And the chip, which I said, it is an electronic device platform that is also being used for the Cas9 for the rapid sensory detection to target the DNA sequence. So this is the one thing which I really wanted to say is that in the first step for a human, what we have to do it is we have to purify whatever the enzymes and the Cas proteins and mRNA guide, whatever the 5 important things we have it also, how to incorporate it in our human body is that we have to first purify our Cas9 proteins. Next, we have to store the Cas9 proteins in a freezer form for the genetic editing coding process. Next, we have to take the human cell, right? We have our Cas9 proteins and we have to freeze it first, and then we have our human cell culture from our, we have to store it in our nitrogen storage only. And then we will plate it in our, what is that, media growth, and we will first separate to take our DNA first with the help of Cas9 and with the help of our media growth plus our DNA. We'll add it all together and we will separate the DNA. We have to first cleave the DNA, right? The mutated one and all. Then after getting the sample at the append of in the end, we'll take it and we have to read it with the help of or incubate it or spin it and remove the media. After that, what we do, we have to electroporate it. What is an electroporate? Through the charge, our DNA is going to wide open its cell and the Cas9 protein is going to go inside our DNA, right, of the cell. So electroporation is really going to happen over here. So electroporation is needed, and then we are going to incubate it in the DNA edit platform. That is nothing but a photo edition is going to see. With that machine, we're going to see how the cell is mutated after the electroporation as the Cas9 goes inside, right? Then we will extract our DNA on a gel. So there in the PCR model, we see the deep sequences, how we get the ATG sequence and all, and there our Pam is going to integrate the mutated things. So like this, we are going to discover a lot of genetic diseases and all. So here is one of the target thing that we have a control of serum over here for the cholesterol. That is this PCSK9 is a gene of a cholesterol where like this is the control and this is the cholesterol patient. So here you can see a rapid down growth over here and rapid up growth over here. This is uninjected and this is the injected one. Just think, we can do the in vivo process, but in XY also many things have been done for this generation to know about the CRISPR and all. In vivo, many things is going to happen. As I said, the Victoria patient is away from her sickle cell anemia, like that. Many diseases are going to be away from the patients or from the humans. So thank you. Hope you all understood.
So very nice and informative talk and webinar delivered by Sophie ma'am. Indeed, it was very good because our students, they are not having that much idea about the new molecular kind of the techniques for the gene editing kind of the mechanism. So, no students, this is open for the asking the questions. So, ma'am,
you can ask.
Yes, we will wait for just 1 minute.
Yeah, okay, fine.
So, ma'am, meanwhile, I just want to ask you that is this CRISPR technology tried with mitochondrial kind of the diseases?
Yeah, they have done it in a first thing they have done it in a muscular dystrophy.
Okay.
In that weakness and all that is done by the CRISPR. In mitochondrial thing, they have done it, but not in humans, of course. That editing process not, the ethical process not done yet. But they have found that the target thing of the mitochondrial thing, like this is there, right? The mutated one, this one, oh god, cytochrome B3, and
Yes.
So in that, it is so dangerous that from one passage to the other passage, we need the carrier of cytochrome B3. So in some patient of a muscular of this mitochondrial thing, the cytochrome B3 is going to be an ineffective way also. So they have targeted this cytochrome B3 and done a study also. But not in an in vivo, it's all in an ex vivo process.
Yes. Yes, but we can hope for the better future.
Yeah, yeah. I can like surely like say not only I, we all the researchers can really say this is like CRISPR can prevent many. We can fight with the viruses and like we can be free with the diseases and all. So CRISPR is really enjoying one
Okay, so
aspect.
how will you see the future of this CRISPR technology in treatment of juvenile diabetes?
Juvenile diabetes. Yes, they
Yes.
have done for the diabetes also. They are going on, but not for the juvenile. I think so. Might be they have done. The papers are there. Like I already said, like hundreds of papers have been releasing on and on.
Okay. Okay.
I have to go through the study.
Okay, but what you said, that example of treatment of the sickle cell anemia, it was really very nice because so many generations they are, you can say that they will be having this kind of the boon that they can give the better bloods to their future generations to come. Okay,
Yes, and
because
mostly in US, everyone, no, I can't say everyone, might be in that 90 percentage, mostly like around 25 percentage of the people are having sickle cell anemia. They are like
yes,
suffering for years and years. Even their
yes,
generation is also having the same.
yes, yes, indeed, it is very useful.
Mm.
So
So that's
that is,
updated one. Feb 8th, it
yes,
has been cleared.
yes, ma'am. That was very nice of you. Hello. 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 Ma'am, are you there? Hello. Hello. Ma'am, are you there? And we have to make them understand every time, right? That you can please wait for some time. This won't be cultivated. Okay. When we are seeing, I think, sir, she has left the meeting, maybe due to some connection problem, or maybe the charge was not there. So, is it okay that we should wind up with this meeting? Hello. 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 So, hello. Yeah, we can have. Can have a series of lectures in which we can ask them to cultivate such values, and we can also take do some workshop type of things. Just a minute, sir. Ma'am is going to join. Right. ये सारे टेस्ट जो हर्षित के फॉलो होने वाले हैं। मैम एक मिनट सर ने तो मना कर दिया है। रिसर्च की बात करते हैं, बेसिक सेक्टर तो चल नहीं रहा है यहाँ पे। रिसर्च नहीं है, सेक्टर तो फिर भी ठीक है, नॉर्मल रेफ्रिजरेटर। परेशान हो गई हो फॉलो अप ले लेके इंस्ट्रूमेंट्स का। हेलो